Hey everybody, Mr. Grove here. Today we're going to talk a little bit about the cell membrane and how we get things across the cell membrane in and out of the cell. So we'll start with what the cell membrane is made up of. And so the first thing is going to be these phospholipids. And so a phospholipid, as we know from chapter 2, is going to be a lipid, which means it's got a glycerol head and then two fatty acid tails. And so this glycerol head part is going to like water, and we would call that hydrophilic. And then these nonpolar fatty acid tails are going to not like water, and so that would be hydrophobic. And so the membrane itself is going to be a double layer of phospholipids. So we've got tail on tail, so a really thick layer of nonpolar. And that's going to come into play with what can get in and out of the membrane. Another thing is going to be cholesterol. And so cholesterols are going to be these little molecules here that are located embedded in the phospholipid membrane. And they're going to help keep from overcrowding and also on kind of a temperature control. So cholesterols are important in that way. Another thing is going to be the carbohydrates. And so on the outside of the cell, attached either to proteins or sometimes attached to the lipids themselves, are going to be called carbohydrates. And so these are, we would call that protein a glycoprotein or a glycolipid, glyco kind of along the lines of carbohydrate. The idea of the carbohydrates is these are your ID badges. And so these are going to help identify this cell belonging to your body. And so if a cell does not belong, then your body sends out the defenses, antibodies, white blood cells, to try and destroy that. And so carbohydrates are the ID badge that, allows your body to identify your cells. And so we're concerned with this in like blood transfusions or organ donors. Is very important to try and match up the carbohydrates of the new cells with that of the recipient. Because if we don't, then those new cells are just going to be destroyed by the recipient's body. So we kind of try and trick them by matching everything up. And so that's why we have like blood types and organ donor matches. The last parts of the cell membrane are going to be all proteins. And so they'll have kind of different jobs. So the transport protein is going to go all the way across the cell membrane, and it usually has some sort of a channel that it can act as kind of a doorway to allow things to get across the membrane. Other types of proteins are receptor proteins, and so sometimes the receptor protein, something will come and kind of hook on the outside, and it doesn't transport the actual molecule across, but what it does, it gives a message that goes across. And then enzymes are also going to be proteins, and so sometimes a substrate molecule will come and interact with the enzyme at the active site, and then that will break down and form the two products then. And so these are all components of the cell membrane. We call our kind of schematic of the cell membrane the fluid mosaic model. And so it's fluid because all of this stuff can kind of move around inside these floating phospholipids. The mosaic part comes from, it's a very large variety of things in the cell membrane. And so that's where we get the mosaic. Now let's kind of concentrate on how do we get things across the membrane, in and out of the cell. We can do that one of two different ways. We can do it passively or actively. And so if we have passive transport, that's going to not require energy by the cell. It's for free. But active transport, the cell's going to have to spend some energy, most likely in the form of ATP, to make that happen. So let's talk about some passive transport first. And so diffusion would be one type of passive transport. So passively, it requires no energy. And this is going to go from a high concentration of particles to a low concentration of particles. That's the direction that the diffusion would take place. So if we have a whole bunch of particles over on this side, the they're going to slowly diffuse across until we reach equality, and we would call that equilibrium. And so that movement from high concentration to low concentration is what we would call a concentration gradient, and it would be what we would say going with the concentration gradient. What kind of molecules can diffuse? If you're really, really small, or if you are really nonpolar, that allows you to diffuse across the membrane. So something like oxygen is very nonpolar. Carbon dioxide would be very nonpolar, and so that can go across the membrane easily, because remember, the majority of the cell membrane is nonpolar from those fatty acid tails. If you're really, really tiny, you can also go across the cell membrane, but you have to be really small. So like water is smaller than oxygen, but it can't really get across on its own because its polarity does not work with the nonpolar part of the cell membrane. So water is going to require these little things called aquaporons to help them get across. So diffusion is one type of passive transport. Another type is going to be osmosis. And so osmosis is basically a special type of diffusion that involves water. And so water can go across, but it has to go through kind of a special part called these aquaporons. It can't just go through the phospholipids. And so the thing with water, it wants to move towards the side of the cell that has a higher concentration of solute particles. So if the outside of the cell is higher in concentration of like salt or sugar particles, we would call that outside of the cell a hypertonic solution. If the outside of the cell was lower in concentration of salt or sugar particles, we would call that a hypotonic solution. And if it was about the same in size, we'd call that an isotonic solution. So hypertonic solutions, the water is going to move towards the sugary, salty. This would be an example of like when we're making pickles. So when you make pickles, you're using a cucumber, which is very watery, so those cells have a lot of water. And then we soak those cucumbers in a real salty, sugary solution, kind of a brine. And the water will then want to go towards that, and that causes the cell to shrivel. So hypertonic solutions will shrivel up the cells, and that could possibly cause the cell to die. That's why you're also not supposed to drink salt water, because you're drinking salty solutions, and the water inside your cells will want to go to the outside where you've been bringing in salty water. So hypertonic, hyper means more than, and so we have more than the solute particles, and so that's why water wants to go towards them. Hypotonic is going to be less than, and so a hypotonic solution, the water would want to go towards the sugary, salty inside the cell. And so if that's the case, that causes the water to go in and the cell to swell up and potentially burst, kind of like a water balloon would if we put too much water in it. And so something like this would be if we're in like freshwater situation. Here was a sad case several years ago when the Nintendo Wii came out, and that was a really hot thing. People were lining up on Black Friday lines, spending the night to try and get their Nintendo Wii. And so this radio station out in Sacramento gave away a Nintendo Wii in this contest that they called Hold Your Wii for a Wii. And so the contestants had to drink water every 15 minutes, and then the contestant that could go the longest without having to use the bathroom won a Nintendo Wii. So kind of a clever play on words, hold your Wii for a Wii. The problem is one of the contestants was consuming so much water that she was creating a hypotonic solution for her cells, and so the water that was pure water that she was consuming wanted to go in towards the sugary, salty part of her cells, and she ended up dying from that. And so it was a tragic incident where this hypotonic solution caused her cells to burst and ended up killing her. Another example would be contractile vacuoles. So if you're a freshwater protist, you have a single cell, and you live in fresh water, so your cells are going to be more sugary. So you basically live in a hypotonic solution. And so the water's constantly coming into your cells, so you've got to figure out a way to pump that out. And that's what the contractile vacuoles do. They squeeze out that excess water back into the environment. So it's kind of like a sump pump in your basement. When it rains and it gets water in your basement, the sump pump will help push that water back out of the basement. So this contractile vacuole will help push the water back out of the cell for these freshwater protists that live in a hypotonic solution. So what we're shooting for usually is isotonic. So we like that same outside-inside environment, and therefore water is not going in too much or leaving the cell too much, but it would stay more at an equilibrium. Another type of passive transport is facilitated diffusion. So in the case of that, facilitated diffusion or ion channels, basically what we're using here is a kind of a transport protein where it's just a doorway. So that really polar molecule that normally could not go through the phospholipid bilayer because of its polarity now has a doorway, and that allows it to go through. We still have to go with the concentration gradient, and we also still have to, and it doesn't cost any energy, it is passive transport, but that is going to require these special proteins to enable these particles to get across. But that's another type of passive transport. Now, active transport is going to be when the cell has to spend energy. And so there's different reasons the cell might want to spend energy. That could be to pumps against the concentration gradient. So it's like when you're paddling upstream in like a kayak or canoe. Maybe it could be that you are going against a electrostatic. So you have positive, negative issues that you have to try and go against. It could be something that we are just going to be too large, and so we have to spend energy to get the particle across the cell membrane that way. So one example is the sodium-potassium pump. And so this is something in your cells that helps keep your sodium and potassium levels at the right stage for electrical transport of nerve signals. And so what your cell does is it pumps sodium ions to the outside of the cell, and then it brings in potassium ions to the inside of the cell. And there's already a lot of sodium on the outside, and there's already a lot of potassium on the inside. So we're going against the concentration gradient. There's also an electrostatic gradient that we have to go against. And so all of this costs energy, and that energy is going to be in the form of ATP. So that's one very common example of active transport is the sodium-potassium pump. Another type of active transport is when we're trying to move really, really large particles. I mean, they're too big to even fit through these little doorways of our transport proteins. And so this is going to be exocytosis and endocytosis. So exocytosis is when we are exporting large particles, probably some sort of waste. And then endocytosis is when we're going to be importing these large particles, probably some sort of food or nutrient. And so the types of particles that we'll do, in the case of endocytosis, this phospholipid bilayer will just kind of pinch off around the particles as they come in, and that will form what's called a vesicle. And then that vesicle, which is made of then phospholipids themselves, can move throughout the cell and take that to wherever it needs to be. But this does require ATP energy to do that. And then on the flip side, if we're doing exocytosis, maybe we have something already in a vesicle it just kind of morphs into the phospholipid bilayer, and then as it opens up, it allows those particles to be released to the outside of the cell. So when we're bringing nutrients in, that's a type of exocytosis. Sometimes we call phagocytosis, that's cellular eating. Or if it's kind of a liquid kind of thing, cellular drinking would be pinocytosis. And so those are also some cases of active transport. So in review, we've got passive transport. We don't need energy. It has to go with the concentration gradient. Examples would be diffusion, or a special kind of diffusion for water, osmosis. Or if we're going through these transport proteins like an ion channel or facilitated diffusion, but that none of those require energy. Active transport, we need energy, so that would be if we're going against the concentration gradient, or if we have really large molecules, or if we are pumping against the concentration gradient like in the sodium-potassium pump, or if we're going to do large exporting and importing of molecules to the cell. And then receptor messenger proteins, also active transport. Sometimes they will require ATP to trigger that message to be sent across. So those are the ways we get things across the cell, and that's the cell membrane, and I hope that was helpful.